Hi everyone, my name is Aaron Marsman with Imhotep's Legacy Academy, and this video will be about the Grade 7 activity 7.1, Why Are We All So Different? This activity is trying to show students the structures and components of deoxyribonucleic acid, or DNA. With all lessons, they start with a proverb and the Peters projection map, which we'll show in a moment. The proverb for this activity is, "Only the knife shows what the inside of the cocoa yam looks like," and it comes from West Africa. So for some of the activities, well, most of them, it'll say a specific country, and then you can use this opportunity to say something about the country, maybe something culturally relevant or something that's happening in the news lately. So what you want is to explain to the students what a proverb is, which is a saying that has some meaning, and then try to get them to share what they think it might mean. So for this one, I could interpret it as you need a specific tool to accomplish certain tasks. But they can interpret it any way, and there's not really any wrong answers with this. But you want to try and connect the proverb with the lesson, and in this case it's DNA. So this is a Peters projection map, and you'll have one when you're doing your presentations with the students. What's special about a Peters projection map is it shows all the countries and continents and land masses in their relative size to each other, so nothing's skewed because of how close or far away it is, like it is on other maps, based on how close it is to the equator. So with this activity, the proverb comes from West Africa, so try to keep it fun. Maybe ask them, where's Africa on the map, what color it is? Does anybody want to come up and point it out? And then where's West Africa? Whatever we would consider West Africa should be the Western countries in Africa. So after you've gone through the intro and the proverb and the Peters projection map, you'll want to do a little bit of background info. Although sometimes you can start certain activities with activity, it's flexible and it's up to you as the mentor. So for the background information for this activity, I drew a diagram of a section of DNA. So you can do it like this and leave it unlabeled and ask the students for what the different types and components are, and you can label it as you go. So like sugar and phosphates, chemical bases, show them what a nucleotide is, and then that 3 nucleotides on the same side of a DNA strand make up a codon. You can talk about how it's in a ladder shape, and that when it's actually looked at, it's a twisted ladder. So get them familiar with the structure and the components of DNA. For the first activity in this presentation, you'll be making edible DNA strands with your students. What you'll need is colored mini marshmallows. Those will represent the chemical bases. Toothpicks, that's what the chemical bases will be stuck to. As you can see here is an example. And then Twizzlers or licorice, which will be the sugar phosphate backbones. So for this activity, you're going to take toothpicks and you're going to take your marshmallows, which represent the chemical bases, and pair them together. So you can give a sequence that you want the students to follow. You can break them up into groups, so maybe 2 students will have a station each. So once you get this activity underway, you're going to take your marshmallows and stick them on the toothpicks, and the marshmallows represent the chemical pairs. So you're going to assign a color to each chemical base. So you have adenine, which only pairs with thymine. So the 2 colors that are assigned to each of those chemical bases can only be paired together. And then you have guanine and cytosine, and they can only be paired together. So you can give a sequence that you want the students to pair, and they can follow that sequence. And you want every student to make their own, so they have something to take home from this activity. So in the end, once you have all the toothpicks with their pairs stuck into the backbone, which is the licorice, then you have your ladder shape and all the components of your DNA strand, and you can twist it, and it takes on the shape of a double helix, and it looks like a strand of DNA that the kids can eat. So you can encourage them to take it home to show it off, but a lot of them will probably be eaten in the classroom. And recap over all the different components and parts of a DNA strand once you've gotten to this stage, just to try and make sure the kids take home some knowledge of the components and parts of a DNA strand. Activity 2 for this presentation is a DNA alias sheet. The end result or product of this activity is a bracelet that will spell the students' names in codons that are color-coded, and they can take that home. So for this activity, you'll need this DNA alias codon table. And as you can see here, each color bead will correspond to a chemical base. Then you'll have your beads, and you see your 4 colors here. You'll have elastic band that you will use to put the beads on and tie off to make a bracelet or necklace, depending on how long the student's name is. And you'll need scissors, and then every student can make their own bracelet for this activity. We're going to take the elastic string, if I can get it out, there. Then I'm going to look at the alias codon table. So here it says adenine is represented by green beads, thymine is represented by red, cytosine is represented by blue, and guanine will be represented by orange. So I'm going to try and spell my name. So the first letter of my name is A. So if I look at the sheet, the simplified codon we have that corresponds to A is guanine, cytosine, and thymine. So guanine is orange, so I'm going to put that on. And then I need cytosine, which is blue, so I'll put that on. And then I need thymine, which is represented by red beads, so I'll put red on. So then, on my string, I have one simplified codon, which represents the letter A, and also on the sheet, every letter represents, and every simplified codon represents an amino acid. So for me, with the letter A here, this represents alanine. So you can make a knot in between each codon if you want. So we'll take our scissors, cut the string. You can knot it at the end, or you can put a hook on the end, which is what I'll do here. So these are what we have here. Just a hook like you see on most necklaces to make it easy for the kids to operate it like a regular piece of jewelry. So we're going to take our end of the string here and release this. So this is a necklace that has the first simplified codon attached, and it has a loop on the end. So after you put on all the simplified codons to represent all the letters of the students' names, then you can tie on the hook on the other end. You can see there. And then you'll have a bracelet or necklace that spells out the students' names and can be opened and closed like a regular piece of jewelry, and they can take that home and show their parents and friends. This is Activity 3, DNA extraction from plants. For this activity, you'll be extracting DNA from a banana. For the first step, you'll need your blender. You'll need 200 milliliters of cold water. You'll need your bananas, 2 to 3, and you'll need 1/8 of a tablespoon of salt. So you'll mix those three things together in the blender and then turn the blender on high for 15 seconds. After you have that stirred, you're going to strain that mixture. So it suggests to use cheesecloth, this is cheesecloth, or a coffee filter to filter it. So if you're using cheesecloth, you want to cut out a square large enough to cover the top of your beaker. You'll put it over the opening, take your elastic band, and just secure it. Then you'll take what you mixed in the blender, which was the salt, banana, and cold water, and then slowly pour it, maybe push down a little bit in the middle because it won't all go through immediately, and then you'll pour it slowly onto the cheesecloth, and then you'll strain out any chunks. They'll stay trapped in the top, and then you'll have the liquid, which is what you'll be working with, in the bottom. You'll then add 2 tablespoons of liquid detergent, and then you'll wait at least 5 minutes, 5 to 10 minutes, for the mixture to sit. Now you're going to take smaller beakers. It depends on how many student groups you have present, and you'll divide up the mixture that you have in the large beaker. And you want to fill this about 1/3 full, so about 35 milliliters. So with the meat tenderizer, you'll add a pinch to each test tube or small beaker, and then you'll instruct the students to stir very gently. Tell them if they stir too hard, it'll break up the DNA, so they won't have strands at the end, which is what we're looking for. Then the last step is to take rubbing alcohol and mix it with food coloring, and you want to have enough rubbing alcohol so it'll create about a 2-inch layer on top of the water. You're going to pour the rubbing alcohol with the food coloring into your beaker or tube. So you have to pour it in slowly, and a trick that helps it get on gently is take a spoon and put it up against, take it and put it upside down and put it against the edge of the beaker. If it's a tube, you can just tilt the tube and pour it in slowly, and then you're going to pour your alcohol slowly onto the top of the spoon and let it trickle down. That way it won't disturb it too much, and you'll be able to create your layers easily and without too much mix. This is activity 4, human DNA extraction. For this activity, you'll be getting DNA out of cheek cells from a student participant's mouth. So, for this activity, you're going to mix 1 tablespoon of salt with 3 tablespoons of water, and you'll have a student who wants to gargle and spit that water back into this cup. So I'll do that for this demonstration. Okay, then you're going to take dish detergent and put it on the tip of a stir stick. So I'm just going to carefully put some on here so I don't get it everywhere. Okay, so you don't want to stir very much because you don't want the water to get frothy. So I'll stir it 3 times really slowly. One, 2, 3. Okay, now you're also going to have rubbing alcohol mixed with food coloring. So you're going to take your spoon upside down, put it against the wall of the cup, and you're going to slowly pour the rubbing alcohol in. So alcohol is less dense than water, so it will create a film on top. Nice and slow. There. So now, at this point, you want to have your students come up close and look from the side, and what should happen is the DNA that would have been broken out of the cells should rise up from the water into the top level, which is the rubbing alcohol. So that's activity 7.1, Why Are We All So Different? You'll be able to get a copy of the activity plan from your coordinator, or you may have received all of the activity plans. Either way, print one off and use that as your guide. At the end of the activity, if you have time left with your students, you can have them do this word puzzle. And it's always fun, I find, to maybe make it a competition or break them up into teams so they're engaged and they find it to be challenging when they're doing this game at the end. Always remember, when students are doing anything, or when you're doing anything in front of the students, encourage them or demand that they wear safety gloves and safety goggles. They're not dealing with anything too dangerous here, but we want to get them in good habits for if they do continue on to a STEM field in university, when they will be dealing with dangerous chemicals. The final activity is activity 4, which is extracting DNA. This can be done for human DNA or for plant DNA. If you'll be doing the human DNA option, restart. This is activity 4, which is DNA extraction. It can be done to get human DNA or to get plant DNA through a banana. If you're doing the human DNA option, you'll be getting spit from a participant. Restart. This is activity 4, DNA extraction. This is activity 4, human DNA extraction. For this activity, you'll be asking a participant to gargle salt water to get DNA cells that are released from their tongue and in their spit. For this activity, you'll have 3 tablespoons of water mixed with 1 tablespoon of salt, and mix it well so it completely dissolves. Then you'll have the student take this in their mouth and gargle it, and then spit it back into the cup. So I'll do that now. Oh, cut. Redo.